the presentation, I want to sort of draw your attention to the banner out here. So tomorrow is going to be the World Suicide Prevention Day, and it's organized by the International Association of Suicide Prevention, and I'm putting up a candle here because there's a campaign where we're encouraged to light a candle near our window at 8 p.m. tomorrow to show our support for suicide prevention, to remember lost loved one, and also for survivors of suicide. So yeah, which is very kind of related to the topic today, which is investigating the etiology of self-harm using genetically informed methods. So, oops, here is the outline of my presentation today. So first, I'll talk about what is self-harm and why do we study self-harm using genetic methods. And then there's these two main studies that were talking two different kind of parts. So the first one is using classical twin design, so kind of like the quantitative genetics part in studying self-harm. And in the second study, kind of more into the molecular genetics part, where we use polygenic scoring and Mendelian randomization. And finally, some general discussion and also future directions. All right, so what is self-harm and why do we study self-harm using genetic methods? So according to the NICE guidelines, define self-harm as any act of self-injury and self-poisoning carried out by an individual regardless of intention or motivation. So we can sort of see self-harm as an umbrella term that can be further refined into suicidal self-harm and non-suicidal self-harm, so i.e. with and without the suicidal intent. And actually, there's an ongoing debate whether non-suicidal and suicidal self-harm should be seen as two separate entities. So on one hand, we have the DSM-5 coming out with non-suicidal self-injury and suicidal behavior disorder as two separate conditions for further study. And each of them has like clear list of definitions, and they kind of mirror the non-suicidal and suicidal self-harm. But on the other hand, we have suicidologists and self-harm researchers objecting this view that suicidal and non-suicidal self-harm may be a false dichotomy, mainly because the motivation behind self-harm can be very fluid. The motivation, so it can vary from episode to episode, or even within a single self-harm episode. And also non-suicidal and suicidal self-harm are highly correlated, and in fact, the best predictor for a suicide attempt or suicidal self-harm is having a history of non-suicidal self-harm. So why do we want to use genetically informed methods to study self-harm? So one of the main motivation was to investigate the etiology of self-harm, like to what extent, so because we know that self-harm runs in families, so to what extent genetics playing a part or environment, and especially we were interested to know if there's any etiological difference between suicidal and also non-suicidal self-harm. And the second motivation is kind of based on kind of like this golden adage that correlation doesn't imply causation. So in many classical epidemiological studies, risk factors for self-harm or suicide has been identified, but because they are associations, and when sometimes this is kind of like a debate whether these correlations are confounded, or is it kind of, do they really imply causation? So yes, genetic studies are kind of based on like associations as well, but we're hoping that with some assumptions that we can use genetic data to help strengthen the causal inference, mainly due to these two main points. So first one is genetic sequence is fixed at birth, so the reverse causation is quite unlikely. And also the genome is randomized at conception. So there's this kind of like idea of randomization kind of mimics the randomized control trial. And for some risk factors, it's quite unlikely that we can conduct a randomized control trial. So for example, if we want to know if smoking is contributing towards self-harm, we cannot assign a group of people to smoke and another group of people not to smoke. So that would be an unethical randomized control trial. But by leveraging, by using genetic data, we can potentially kind of exploit that, that randomization part. But firstly, we're going to talk about some twins, like classical twin design first. Classical twin design is based on the fact that we have two types of twins: the monozygotic twins, which is a fertilized egg. So one egg fertilized by one sperm split into two, and dizygotic twins or DZAC twins, which are two separate eggs fertilized by two different sperms. So for monozygotic twins, they share 100% of their segregated genes, and for dizygotic twins, they share 50% of their segregated genes. And using these genetic similarities between the MZ and DZ twins, we can decompose the variance for a phenotype or covariance between two phenotypes into genetic and environmental components. So we can decompose the variance of these phenotypes into A, which is additive genetic component, C, which is shared environmental influences, which means environmental influences that make a twin pair more similar to each, so make two twins more similar with each other. And E, which stands for non-shared environmental influences or unique environmental influences that make the twins different from one another. So as you can see, there's this genetic relationship of 1 for monozygotic twins for the additive genetic influences, and 0.5 for the dizygotic twins. Whereas for the environment, we for both the MZ and DZ twins, we assume there's this correlation of 1 for their shared environment. So this is based on this assumption called equal environment assumption, in which we assume that both MZ and DZ twins share this environment with a correlation of 1. So now I'm going to talk about using classical twin design to study self-harm. A little bit of background. Previous twin studies mainly focus on suicidal self-harm, and in a review for a broader phenotype of suicidality, which include suicidal thoughts, plans, and suicidal self-harm, suicide attempt itself, the heritability is estimated to be up to 55%. And when I try to look for any published twin study that look into kind of like difference or correlation between non-suicidal and suicidal self-harm, the closest one that I found was an Australian twin study based that they look into non-suicidal self-harm versus suicidal ideation. So the suicidal ideation is kind of like a proxy for the suicide attempt itself. And as you can see from this diagram, non-suicide, so that's like a strong genetic correlation between non-suicidal self-harm, in this case it's non-suicidal self-injury, with suicidal ideations of about 0.95 for males and 0.66 for the females. And for the non-shared environmental correlation, it's just kind of lower, it's 0.2 for the males and 0.54 for the females. But this is suicidal ideation, but not suicidal self-harm itself. So we conducted study 1 with the question in mind: is there an etiological difference between non-suicidal and suicidal self-harm? And there's also like two aims. So the first aim was to investigate to what extent suicidal and non-suicidal self-harm share the same genetic and environmental etiologies. And the second aim was to investigate the shared genetic and environmental overlap between a range of mental health measures with both suicidal and also non-suicidal self-harm. We used data from the twins' early development study, where self-harm data are available for about 9,000 twins. The self-harm measures were collected at age 21, whereas the mental health measures were collected at age 16. So there were 90 mental health measure questionnaires in total. Some were self-reported, some were parent-reported, some were both self and also parent-reported. And here is kind of like a glimpse for the self-harm questionnaire that the twins answered. So for non-suicidal self-harm, the twins were asked, in your lifetime, have you ever hurt yourself on purpose without intending to kill yourself? And they could either answer no, never in my lifetime, or yes, with different levels of frequencies. And for suicidal self-harm, the question was, in your lifetime, on any of the occasions you have hurt yourself on purpose, have you ever seriously wanted to kill yourself, with the similar options available? So here's some descriptive statistics. For non-suicidal self-harm, about 22% of the twins reported to have harmed themselves at least once in their lifetime without the intention of killing themselves. However, for suicidal self-harm, the prevalence is slightly lower, but also quite alarming. So about 10 to 11% of the twins reported to have harmed themselves with the intention to kill themselves at least once in a lifetime. Here is the list of analysis that we carry out. We use liability threshold models because the variables were ordinal variables, so we sort of had to assume that there's an underlying normally distributed liability for suicidal and also non-suicidal self-harm. And there are three types of models. So you have univariate models to estimate the A, C, and E components for suicidal and non-suicidal self-harm separately. So just to recap, A is the additive genetic influences, C is the shared environmental influences, and E is the non-shared environmental influences. And then we look at the correlation between suicidal and non-suicidal self-harm, and we conducted a bivariate model to estimate to what extent genetic and environmental influences are there for the correlation between these two phenotypes. So that's to estimate the genetic and shared and non-shared environmental correlations between suicidal and non-suicidal self-harm. And we also conducted like a formal test, a goodness of fit test, to test whether these two phenotypes can have different etiologies. And finally, we conducted a series of trivariate models to estimate the genetic and environmental correlations between each mental health measure with both the suicidal and non-suicidal self-harm. It probably will become a bit clearer when I show the diagram later on. But we, similar to the bivariate model, we also conducted the goodness of fit test to test whether suicidal and non-suicidal self-harm can be different in their etiological relationships with these mental health measures. And through all analysis, we used we included age and sex as covariates in the models. So here are the results for the univariate models. You can see that the glass is sort of half full, so the heritabilities for both non-suicidal and suicidal self-harm are about 50%, with another 50% accounted for by non-shared environmental factors. And the shared environmental influences were estimated to be 0. Here is the result from the bivariate model. There's a strong phenotypic correlation between suicidal and non-suicidal self-harm, which is 0.87. And if you look at the genetic correlation here, it's very close to 1 between suicidal and non-suicidal self-harm, which is 0.94. The non-shared environmental correlation is 0.8, whereas the shared environmental correlation was estimated to be not significant, which is understandable because if you recall from the univariate model, the shared environmental influences were estimated to be 0. So to sort of summarize it, 56.3% of the covariation or correlation between suicidal and non-suicidal self-harm can be explained by genetic factors, whereas the rest of the correlations, is 43.7%, can be explained by the non-shared environmental factors. And then we tried to carry out a goodness fit test. So what we did was we constrained the A paths here to be the same, and we also constrained the C paths here to be the same, and the E paths here to be the same. So essentially we're asking the question, in this bivariate model, can suicidal and non-suicidal self-harm have the same heritability, the shared environmental components, and also non-shared environmental components, like in terms of the estimates. So we constrained the 3 pairs of paths estimates to be the same, and we compared it against the non-constrained model. The p-value was not significant, suggesting that the constrained model is not a worse model than the non-constrained model. So this kind of evidence that suggests that there's no etiological difference between suicidal and also non-suicidal self-harm. But how about when we consider them together with the mental health measures? So this is why we carry out the trivariate models, and it's essentially an extension from the bivariate model. So these two are kind of like the bivariate model. We have the non-suicidal and suicidal self-harm, and then we just add in one more mental health measure. And the list of mental health measures, there are 19 of them, but in general, they can be sort of categorized into 5 groups. So we have internalizing problems, externalizing problems, psychotic-like experiences, substance abuse, and also others like the total mental health difficulties indexed by the strength and difficulties questionnaire, and also a questionnaire for autistic traits. So the question we ask in the trivariate models is whether, in consideration of mental health measures, there like a different etiological relationship between suicidal and non-suicidal self-harm with the mental health measures. So in this example, we're asking the question, is the genetic correlation between this SDQ with non-suicidal self-harm the same as the genetic correlation between SDQ with suicidal self-harm? And it's not just for genetic correlation, but also for shared environmental correlation and the non-shared environmental correlation. So we did this constraint model and compare against the non-constraint model, again, like in a goodness of fit test. So the question was, are non-suicidal and suicidal self-harm different in their etiological relationships with the mental health measures? And we did this test across all these 19 mental health measures. And after all the analysis, the answer is no. This is probably not very surprising, given that in the bivariate model, we found that they have strong genetic correlation, and also from the goodness of fit test, there's no evidence of etiological difference between suicidal and non-suicidal self-harm. So that's kind of what we did in the first study. So we did not find any evidence of etiological difference between these two types of self-harm. We found a strong genetic and environmental correlation, suggesting that there may be a similar biological mechanisms and unique environmental influences behind these two phenotypes. But of course, while the main limitation is the definition of non-suicidal and suicidal self-harm in the current study, they are not the same as defined by the DSM-5. So for example, in the DSM-5, non-suicidal self-injury is defined as damage to the surface of the body. So that would not include non-suicidal self-poisoning, which is kind of being included in the definition for non-suicidal self-harm itself. But at this point, I probably will ask the question of why is DSM-5 not including non-suicidal self-poisoning in the definition for non-suicidal self-injury? Perhaps some of you here can probably know the answer, but it's kind of something that kind of bothers me sometimes, because I have also heard from other conferences and presentations that people who engage in non-suicidal self-poisoning, when they were admitted to the A&E, they're being coded as a suicide attempt, but in fact, they ingested they self-poisoned without the intention to end their life. So that's something quite interesting. So now we're going into the second study, which is more like the molecular genetics part, using polygenic scoring and Mendelian randomization to study self-harm. And this is based on the study has been published recently on PLOS Medicine with the title Studying Individual Risk Factors for Self-Harm in the UK Biobank using polygenic scoring and Mendelian randomization. So the aims were twofold as well. The main aim was to identify plausible causal risk factors for self-harm, mainly using the Mendelian randomization method. And a more kind of secondary aim and sort of like connected to study one is to investigate if there is etiological difference between non-suicidal and suicidal self-harm. And that's mainly using the polygenic scoring method. So we use data from the UK Biobank, which is the largest biobank in the UK and probably one of the largest in the world as well, with about 500,000 participants. But when we zoom into self-harm and genetic data itself, the final sample size was about 126,000 participants, a little bit more females, and their ages range from 48 to 82 years. So we have to keep in mind that they're mostly middle age or at a more advanced age. So there's no participants in their 20s or 30s or even younger. And the self-harm measures were self-reported as well and was collected from the mental health questionnaire administered to the UK Biobank participants. And here's the question asked for self-harm. In order to ascertain whether the participants have harmed themselves, the question we use is this, which is, have you deliberately harmed yourself, whether or not you meant to end your life? And here's this flow diagram. So out of about 126,000 participants who had the genetic data, when they're asked this question, some of them say they prefer not to answer. So we end up with this final sample, and out of this final sample, about 4% of the participants responded that they have deliberately harmed themselves, whether or not they meant to end their life. So that's about 5,000 of them. And to ascertain whether they engage in suicidal or non-suicidal self-harm, a second question was asked for those who responded that have self-harm previously. So they would ask, have you harmed yourself with the intention to end your life? And again, they could either choose yes, no, or prefer not to answer. And as you can see, the distribution is kind of like half-half. So about 2,800 people answered yes, which means they self-harm with suicidal intention, and those who answered no were ascertained as those who self-harm without suicidal intention. You can probably see a potential problem here. So back then in study one, the participants were allowed to answer they have either engaged in suicidal and non-suicidal self-harm in their lifetime. So it wasn't a forced dichotomy. So in this question, for those who have engaged in suicidal self-harm, there is a possibility that they have also engaged in non-suicidal self-harm, but because there is no way to know that, I hope it makes sense. So from this question, have you harmed yourself with intention to end your life? With the responses of yes and no, I can only know if the participants have engaged in suicidal self-harm. And I'm not so sure from this group of people how many of them has engaged in non-suicidal self-harm. So that's kind of like a potential limitation for the study. So here are the analyses that are being done. So we carry out polygenic scoring, asking the question, do polygenic scores of different risk factors predict self-harm? And if they do they predict suicidal and non-suicidal self-harm differently? And building on top of the insights we gain from polygenic scoring, for those risk factors with polygenic scoring significantly predicting self-harm, can evidence of plausible causal relationship be found using this method called Mendelian randomization? So a little bit of intuition about polygenic scores. A textbook definition for polygenic score is a single individual level score computed for a given trait, weighted using summary statistics from an independent genome-wide association study, or GWAS, for that particular trait. So in order to generate polygenic scores, we need two samples. So the first sample is called the discovery sample, where the GWAS was conducted, and it provides the summary statistics that inform us about the effect sizes of each genetic variant. And the second sample is called the target sample, and in this case is our UK Biobank sample. And each of these participants provided their DNA information so that we can use the DNA information and also the summary statistics from the GWAS to calculate the polygenic scores. And these polygenic scores will be calculated for each of the individual in the target sample, and it will inform the genetic propensity for a particular condition. So if we use GWAS for schizophrenia, we'll be able to generate polygenic scores for schizophrenia, which informs the genetic propensity for schizophrenia. So in this study, we are seeing polygenic score as a genetic proxy for our risk factors for self-harm. So just to sort of illustrate, if major depressive disorder is causally related to self-harm, then a polygenic score for MDD, which is major depressive disorder, should also be associated with self-harm. So through this pathway, because the polygenic score will increase the genetic risk for MDD and subsequently increases the risk for self-harm. And we will see an association between this polygenic score for MDD with self-harm. So we generated polygenic scores of 24 risk factors for self-harm that come from different domains. So we have mental health, we have polygenic scores for mental health conditions, risk factors for cognitive ability, personality traits, substance use, and physical traits. So we try to do it in a systematic way. So we go through all the GWASs with publicly available summary statistics, and then we look at GWASs whose phenotypes are risk factors with reported association with self-harm or suicide from classical epidemiological studies. We also apply some selection criteria. So we only pick GWASs with participants of European ancestry because the UK Biobank is mainly white British, so the ancestries has to be matched. We do not include any GWAS that has UK Biobank participants in it because we need to avoid overlap between discovery sample and the target sample, and because the target sample is the UK Biobank sample, so we have to exclude any GWASs that has UK Biobank participants in it. On rare occasions, wherever it's possible, we try to rerun the GWAS without the UK Biobank participants, so that was possible for the MDD GWAS. And also, we applied a fairly arbitrary threshold. We only included GWASs with a sample size that has more than 15,000 participants, so that we can limit the use of underpowered GWAS, which will in turn become underpowered polygenic scores. So for case control studies, we use the formula to calculate the effective sample size and also apply the same sample size threshold as well. So here are the results for the polygenic score analysis. So here are the 24 risk factors that we use to generate the polygenic scores and also their effect sizes in predicting self-harm. So we will look at the blue lines first, the blue dots, the blue triangle, which is a single polygenic score regression. And because we're running a lot of tests, so we had to carry out multiple testing correction. So after multiple testing correction, only these polygenic scores in a blue box are still significantly predicting self-harm. And we're going to zoom into that. So this 6 polygenic scores the polygenic scores are still significantly predicting self-harm after the FDR correction, which is a way of correcting for multiple testing. And as you can see, they are polygenic scores for MDD, schizophrenia, ADHD, bipolar disorder, alcohol dependence disorder, and lifetime cannabis use. And as a single polygenic score, so kind of like in separate regression models, they are significantly predicting self-harm. And then we wondered, how about if we put all of them in the same regression model, kind of like a multivariate regression? What will happen if we control for the effects of other polygenic scores? And this is when we look at the green lines and green dots, and here are the effect sizes, and we realize that polygenic score for alcohol dependence disorder become not significantly predicting self-harm anymore in the UK Biobank sample after we take into account the effects of other polygenic scores. So these are how polygenic scores are predicting self-harm. Self-harm as the umbrella term, not self-harm in terms of suicidal and non-suicidal self-harm. So how about for these two types of self-harm then? Do polygenic scores for these different risk factors predict them differently? So what we did was we run other tests in multinomial logistic regression models, and we found that none of the 24 polygenic scores significantly distinguish between suicidal and non-suicidal self-harm after multiple testing correction. Okay, so we have identified 5 risk factors, which are mainly from the mental health domain, in which the polygenic scores significantly predict self-harm. But there's some limitation of using polygenic scores in causal inference, mainly because the polygenic score method is prone to unmediated or horizontal pleiotropy, because we tend to use a less significant p-values threshold in selecting the genetic variants to be included in a calculation of polygenic scores. So in this case, we use p less than 0.3 as the threshold in selecting the SNPs. So that will pose a risk of pleiotropy. And fun fact is, in Greek, it means is derived from pleion, which means more, and also tropos, which means way. So instead of going through major depressive disorder from genetic risk of major depressive disorder and subsequently into self-harm, it's going through probably going through a different way into self-harm. So we have to be cautious with it. And this is a time when we use Mendelian randomization, which is a more stringent method to control for pleiotropy. So there are 3 main assumptions in conducting Mendelian randomization. The concept behind it is, imagine we have exposure, in this case MDD, and also outcome, which is self-harm, and we're interested to know what's the so-called causal estimate from the exposure to the outcome, and there's a risk of it being confounded by lurking variables. And the instrument genetic variables kind of become a proxy for the exposure, so that we can know whether it's kind of going into self-harm. And then but these are built based on 3 different assumptions. So the first assumption is relevance, in which the instruments has to be robustly associated with exposure. So in this case, we have to choose genetic variants which are significantly associated with the exposure. Typically, we use genetic variants that pass the genome-wide significance threshold. And the second assumption is exchangeability, which is the confounders do not confound the SNP outcome association. And the third assumption is the exclusion restriction criterion, in which the only pathway of SNP outcome association must be through the exposure. So we would observe an association between the instrument genetic variants with the outcome, which is self-harm, but we are assuming that this association is due to the pathway that it travels through the exposure, then go subsequently into the outcome. I hope this is clear. We conducted 2-sample Mendelian randomization because we used 2 different GWAS which were conducted in 2 different samples. And we will have a GWAS for exposure, which is kind of seen in the relevance pathway here, where we can observe what's the effect sizes of the genetic variants on exposure. And we also need the GWAS, the summary statistics of GWAS for the outcome, which is GWAS for the self-harm. But then the association that we observe in that GWAS is being assumed to have been kind of like mediated by the exposure. So we apply MR analysis on the risk factors that were found to be significant in a polygenic score analysis. And in selecting the genetic instruments for the exposures, we used a p-value threshold of 10 to the power of -5. We did not use the genome-wide significance threshold because for some of the psychiatric, for some of the GWASs for psychiatric conditions, not many genetic variants actually pass through the genome-wide significance threshold. So we have to use kind of like a less stringent p-value threshold. And the result of using this is resulting in a weak instrument bias, which will kind of make the MR estimate a bit more conservative. But we try to sort of address this by running some sensitivity analysis as well. So we use the inverse variance weighted method as the main method for the MR analysis, and we ran MR reps, weighted median, and MR agar as sensitivity analysis to check the violation of MR assumptions. So a little bit more about these different MR methods that we carry out. So the inverse variance weighting, the IVW method, is the most powerful method, but it cannot account for directional pleiotropy. On the other hand, the MR reps, or the robust adjusted profile score method, was used to account for the selection of weak instruments. So in this case, the fact that we selected instruments which are not genome-wide significant. And then the weighted median method, which is more robust to directional pleiotropy than the IVW method, and also more robust to individual genetic variants with outlying causal estimates than the IVW and MR agar regression. This is a case this is because we're using a weighted median rather than a weighted mean, because median is less likely to be skewed by outliers. And finally, we had the MR agar regression method, in which the significance of the intercept term using the MR agar regression will inform us on the presence of directional pleiotropy. And here are the results from the MR analysis. So I just want to draw attention to the MR agar regression in terms of the intercept term here. So the green boxes would take mean that the estimates are significant with p-value less than 0.05, whereas the red boxes with crosses mean they're not significant. So in this case, for the intercept of MR agar, if there's a green box with tick, it means that that is the presence of pleiotropy. So in this case, we observe pleiotropy for MDD, whereas for ADHD and schizophrenia, we have MR estimates that were significant across different methods, but not for the intercept term of the MR agar regression. So suggesting that there might be no risk of pleiotropy for ADHD and also schizophrenia as an exposure or risk factor for self-harm. I just want to talk a little bit more about the presence of pleiotropy for MDD. When I was in a conference, someone looked at my results and say that, oh, maybe this is kind of like a good negative control, because the diagnostic criteria for MDD, which is major depressive disorder, one of them is actually suicidality, which is very related to self-harm. So when you're running a GWAS for MDD, and if it includes suicidality, so it will sort of inevitably capture the genetic influence for self-harm as well. So probably it's a good thing that we observe pleiotropy for MDD in this case. So here are a discussion. So out of 24 risk factors, those in the mental health domain are most plausible to be causal risk factors for self-harm. And this is a case for schizophrenia, ADHD, and also some evidence for pleiotrope for MDD as well, but it has like significant MR estimates in other methods. There's no evidence for marked differences in how the polygenic scores predicted suicidal and non-suicidal self-harm differently. And the fact that we observe significant MR eager intercept shows that there's pleiotropy for MDD. So it's probably the measurement issue of how we define MDD. And perhaps if it's possible, we can run another GWAS for depressive symptoms, but excluding the suicidality part. And then we can rerun the MR and see what happens. But of course, this study is not short of limitations, especially in terms of how we select the GWASs, because we try to avoid having the discovery samples to the UK Biobank sample. And as we know, UK Biobank sample is kind of like a huge addition for sample sizes of most of the modern, more modern GWASs that we have nowadays. So we end up using mainly older GWASs and unavoidably with reduced statistical power, so that when we observe that polygenic scores for certain risk factors are not significantly predicting self-harm, it may be just due to lack of statistical power and also due to this step, some important risk factors were not included. So for example, the GWAS for anorexia nervosa, I think it has a sample size of less than 15,000 participants, so we had to exclude it from our analysis. Also for the UK Biobank sample, they are older, more educated, wealthier and healthier, so they're not fully representative of the UK population. And because they're in a middle age, and when the questionnaire was administered, it was kind of like a, have you ever done that? Have you ever harmed yourself? So it might potentially introduce some retrospective bias. And also finally for the MR analysis, although we carry out some sensitivity analysis, there's still a risk of pleiotropy. So even though the intercept term for the MR aggregate regression is not significant for ADHD and schizophrenia, it doesn't mean that we are 100% sure there's no pleiotropy. So final part of my presentation, some general discussion and future directions. So in both study 1 and study 2, we found no evidence of etiological difference between suicidal and non-suicidal self-harm. And going back to the debate, does this mean that it's a false dichotomy? So does it mean that it's the best if we perceive suicidal and non-suicidal self-harm to be on the same continuum with these two different phenotypes at different ends of the continuum? And also, as we found strong genetic and also non-shared environmental correlations between suicidal and non-suicidal self-harm, and also polygenic scores analysis show no differences in how they predict these two phenotypes. It suggests some opportunities to prevent both with similar preventative interventions. And in study 2, using Mendelian randomization, we found that psychiatric conditions are found to be the most plausible causal risk factors for self-harm. So potentially detection and treatment of symptoms of ADHD and schizophrenia like psychotic or impulsivity symptoms may benefit service users who self-harm. And so this is a slide for the future directions. Actually, it would be great if someone in this group have some comments or ideas as well. So so for my next project, should I focus on self-harm as kind of like an umbrella term, or should I still kind of try to look for etiological difference between suicidal and non-suicidal self-harm? And from a discussion with my supervisors, we are planning to merge methods in study 1 and study 2. So merging twin study, which is the directional causation design, with Mendelian randomization, which is called MLDoc, that was introduced a few years before. And because The target sample for study 2 was UK Biobank sample. We had to exclude a lot of GWASs, so it probably would be a good idea to replicate study 2 in a non-UK Biobank sample so that we can have more powerful GWASs, and preferably carrying out in a younger population, because self-harm is more prevalent among younger people. And the hot topic of intergenerational transmission, should we apply it in studying self-harm and suicidal behavior? And of course, I welcome any suggestions or comments for future directions. So my acknowledgements to my supervisors, John Baptiste and Fruling, who are my PhD supervisor, and Catherine, who supervised my master's project for the polygenic scoring part, and then kind of brought it into PhD and do the Mendelian randomization part. My collaborators from study 1 and study 2, the participants from UK Biobank and also TEDS, and also the people who work hard in collecting the data. And finally, I want to put out some resources for self-harm and also suicide. If you're struggling with things and you wish to talk about it, or you know someone who is struggling, so there's this free line by Samaritans. It's just open 24 hours a day.